No tengo la elocuencia del doctor Horacio Lara para presentar al siguiente ponente, pero les voy a decir en 2 líneas. Para mí es un gran honor presentarlo y creo que para AMVEC también. La mayoría de ustedes estoy seguro que lo conocen, los famosos 10 puntos de Madec. Simplemente les diré que el doctor tiene maestría y doctorado en Francia, fue director de un instituto en Bretaña, que es la zona más importante porcícola en Francia, y ahí tuvo a su cargo un laboratorio de investigaciones veterinarias con 210 personas a su cargo, de las cuales 70 son científicos. Entonces, para que se den un poco una idea de su currículum, tiene maestría en ciencia animal y doctorado en desórdenes reproductivos. Trabajó en el Instituto Pasteur y en la Escuela de Alfort y tiene numerosos reconocimientos en Europa, en Asia y en América. Ahí está el título de su ponencia y lo dejo para no robar tiempo.
Thank you, Mr. President. Buenos días a todos. Unfortunately, these are the only words I know in Spanish. And I'm sorry for that. My Spanish is too poor and I have to speak in English. I would like to start with giving my best congratulations to the organizers of AMVEC Congress. You did a wonderful job. Secondly, I would like to thank the people of 2 companies, the Impexreco company from Brazil and the FEPAC company from here from Mexico in Querétaro, for giving me the possibility to be here with you today. After that, let's start with my presentation. I'm due to speak about the emergence of diseases in farm animals with a focus on pigs. So, the definition of emergence start with the etymology. To emerge come from Latin emergere, to come up, and initially it was to come up from water. It also means to appear, to surface, to come to light. As for health sciences, the main idea is novelty in the thing per se, in the disease per se, or in its development and spread or incidence of severity. So there is an official definition, so you can find this idea of novelty the first time it occurs, but you can also have re-emerging diseases, and in that case, the disease disappeared temporarily from a territory and it came again. Disease emergent has to be distinguished from an emerging infection. You can have an infection without any disease. The pathogen can be discovered without any apparent clinical signs. So this is a sort of slight difference in between the 2. Here are listed many diseases which are were new, considered as new at least. Many of them are reported here. You have pseudorabies in 1974. You have different strain of swine influenza. In 96, we were the first with PMWS, despite the virus was found many years before. And then, of course, elsewhere, you have PED in North America in 2013. So, we can try to sort the emerging health problems into different categories. First, actually new diseases, like PRRS, 1987 in the USA, or PMWS in 96 in Europe. It means that the diseases as such, the clinical signs, the lesions, typical of the disease, were actually new. Then you have new disease to your own country or region. It means that the disease was already present in other places in the world, and it came to your own region or country. We had many cases that can serve as examples of this. Then you have re-emerged after a period of absence. So we have here the case of TGE. We had many cases of TGE, very acute cases, a terrible disease in the 70s in Europe, like that you have now with PED, clearly the same symptoms with the same disasters. And suddenly, in 1984, TGE disappeared by a mere chance, but it was not a mere chance, because we had a new virus coming in. This was the porcine respiratory coronavirus in 1984, and the virus spread very rapidly throughout the world, let's say, and it well protected against TGE. So it was a sort of natural vaccine, okay? So we were lucky. The disease may be always there, but we had not the diagnostic capability at that time, like what we called grain poisoning, and later on we could see that this was due to mycotoxins. Then we have new public health issues, or perception at least, that draw attention on different points like drug resistance, or public fear, salmonella, hepatitis E, emissions from piggeries, more and more, water pollution. And to me, as in the audience, mostly veterinarians, I have the impression that more and more in the coming decades, in the future, the veterinarian will have to not only to take care of animal health, but also to veterinary public health. These are very, very sensitive issues. And the public, we are now with the mediatization, the television and YouTube and other system of communication, the news goes very fast and they can be amplified in a non-reasonable way sometimes. So it is also important, always important to maintain some scientific aspects in the information which are spread. So now we arrive to the reasons for disease emergence. They are multiple. And an important one is the role of movement, translocation, changing in the location of the pathogen, in particular in case of infectious diseases, of course. We had several historical events like the plague of Athens, 400 years before Christ. At the time, the city of Athens was fighting hard against the neighboring city of Sparta. And nearly 30% of the people died on this occasion. Then we had the Black Death in Asia and Europe. We believe, or historical retrospective studies say that the disease came from central arid areas of Asia. It moved to the Mediterranean basin following the Silk Road, and then it moved up to Europe, north, west, and east until Russia. And at that time, the population of the world was around 500 million people, okay? And it came down, according to the historical data, to around 350 million people. So it means that a reduction of nearly 25% of the population of the world. You can imagine the apocalyptic situation at the time. Then we have other diseases. We are concerned here in Mexico with smallpox. Pathogen of biological pollution. Introduction, this was more recently, of course, of zebra into Spain, and with them came a horse sickness. It's an orbivirus that can affect the mules, the equids, and the donkeys, and so on, and the zebra. So the current world looks like a global village with intense trade of goods, different sort of goods, and with them come, we like it or not, additional substances, additional things, flora with the weeds, bacteria, spores of fungi, viruses, insects, okay? So we are in this world, an open world, so necessarily we are in a risky situation, so we have to be careful. So after the movement, translocation, the role of the wildlife, the zoonotic aspects. So about 60% of 300 disease events recorded in humans over the last 60 years were caused by zoonotic disease agents. It means pathogens that can survive in animals and man. And 72% of these originated from the wildlife. The wildlife is a reservoir, but we need a contact with it. And we also need, usually, amplifiers to determine the magnitude of the events, and these amplifiers are usually arthropods, insects, very often. So third point, the role of intensification. And once again, the definition, intensification per se means an intense use of the resources, the buildings, the animals, the money invested, okay? So intensification means a more intense use of the resource. We try to reduce the cycle of the cells, early weaning, okay, and so on. We are asking more to the animals. Then we have accompanying trends like increased size of the production units, specialization of farms or regions. Increased size is the fact that we try to take advantage of economies of scale, which is totally understandable. Then a movement on the long distances. So I think that intensification tends to increase the scale of different things. The pathogen load, when you have on the same place a lot of animals, the same species, it means that the pathogen, when it will enter that, in particular if it is fully susceptible without any antibody, any protection at all specific to this pathogen, it will have free hands to replicate massively. And then, from a given farm to spread to other farms in the neighborhood. The role of global warming. This is a general issue. Now this theory is more and more accepted, I think. We still have some people that don't believe in that, but we like it or not again, but we have believers and non-believers. So the consequences probably are not important in the sense of emergent per se. It could be more important probably in the spread of the disease. All in all, global warming is thought to impact more the distribution of the diseases than emergent per se. That is a point of view of most of the specialists today, especially at the WHO and at FAO. So the role of humans and socio-economic aspects and governance. Pollution, man-made problems, and we will see later that an important part of the problems we have are man-made problems. In relation with compound technologies, negative side effect, unwanted effects, of course, they are unwanted, of genetic engineering and so on and so on. We may have some troubles ahead of us due to a bad control of the application of these new technologies we have now in our hands. We have to be very careful. Changes, disruption in the environment, enveloping the pigs, the diet ingredients, for example, breakdown of efficient disease monitoring that can happen in certain countries, outstripping of community services like potable water, sewage disposal, accident, bioterrorism, deliberate immersion in that case, of course. So now we will go through a number of case studies. We will start with PRRS. You know this disease is by heart, so I will not insist on that. Premature farrowing, cyanosis, respiratory symptoms. Here is a scheme of the virus, which is well known. And a point is that the virus is rather unstable. It means that it is susceptible to changes. And these changes might go along with different virulence. So this virus is susceptible to mutation and to recombinations. Why did it emerge? We have a number of hypotheses based on historical data and recent advances in sequencing and analyzing of the genome of stored isolates. We have a lot of isolates in our freezers all around the world in the veterinary institutes or in virus banks. So now with the new technologies, we can analyze the sequences of those isolates. And it came out that PRRS virus, especially the EU genotype, circulated in Eastern Europe far before the description in the EU. The first description in the EU was in 1990, but far before, the virus was present in the eastern part of Europe. In Europe at large, considerable events occurred since the Second World War. For example, a drop of pea production in Russia, then restoration and construction of large units, and very rapidly. You know that it is far away from you, but we had in Europe so-called the Cold War. It was not in Europe, it was for the entire world. So we had a border all around here, and we could not exchange people on both sides of the Iron Curtain. And in 1989, this Iron Curtain that divided Europe fell down. So suddenly we had a lot of movement of animals, in particular here in Germany, between the western part and the eastern part, German Democratic Republic. Okay? Since the virus was already present for many years on this side, then it came here and it met there totally susceptible animals. So it took the opportunity. The pathogen, most of them are opportunists. So they take advantage of a favorable situation to replicate and then to give disease. So it is believed that translocation, mixing pigs of different health status was the cause of the emergence of PRRS in Europe. The cause of the emergence in North America, in the USA in 1987 is still unknown. The viruses suddenly appear nearly the same time, 87 and 90, and giving the same symptoms, more or less the same symptoms, but the viruses are rather different to some extent, 67% similarity only. So the case of swine influenza, the pandemic we had, just to mention, you know this by heart, that the water birds, particularly the ducks, are a central point, epidemiological point of this disease, epidemic disease. Then you have the domestic birds, you have the humans, you have the pigs, and you have also other animals that can be concerned. The virus, the particularity of the virus, its RNA virus, is that the genome is split into segments. And the second is that, as most of the RNA viruses, they are unstable viruses. So they can change. In particular, they can have some drift change in amino acid sequences of the hemagglutinin, So if I go back, hemagglutinin is a protein here at the surface of the virus, okay? And this is an enzyme. This enzyme is used, particularly with the virus, to enter in the host cells at the epithelium of the respiratory tract. On the other hand, the neuraminidase, N, these ones, are used mainly when the variants are going out from the infected cells. Okay? And these two enzymes, the two proteins, are used to qualify, let's say, to qualify or to rank, to sort the viruses, H1N1, depending on the shape of these proteins. So back, then there are unstable viruses. You can have drift or shift. Shift means reaspawnement or genomic segment. So entire segments of viruses. When you have co-infection, you have two viruses or three viruses at the same time infected an animal. Then when the new variants are being, when at the stage of budding, when they are reconstructing the virus within the infected cells, some segments of different viruses can join and make a new variant, giving a new virus that can be not recognized by the body, and then the disease, if there is no antibodies against it, and then this can give birth to a new disease. So I tried here to mention a hypothesis of the history of the virus, of the pandemic of 2009 in Mexico. So it is thought, we are not 100% sure, but we had a thesis looking at that, and there's a sort of consensus on this. In human 2009, around 2009, so it seems that some segments, some genes were coming from one pig, and other ones, two ones, were coming from another pig, different pigs. So this strain is 100% pork produced, okay? And then, probably around 1998, this pig was infected by a virus, a triple reassortment, with one gene coming from humans, two for ducks, and the other ones from another pig. Okay? And the human himself was infected by duck. On the other hand, this pig kept the entire virus from ducks in around 1979. So this is the hypothesis, retrospectively, giving the history of this pandemic. So the case of PED in the USA, 2013. So this disease is recognized for nearly 40 years, but the USA were free. The description started in the EU in late 1978, but the disease had very moderate economic consequences. It's very surprising. At that time we had both. We had PED and TGE, but PED was nearly nothing. Let's say some mild diarrhea without any consequences, any severe consequences. The problem was TGE. And from that date, PED in Europe declined, and it was not a real problem. And still, I cross a finger, but to me, I don't know, tomorrow when I will go home, maybe we'll have a case, but we don't know. We will see what will happen. In terms of epidemic diarrhea, nothing is definitely established. Nothing is definitely established. We're working with living microorganisms with a very rapid cycle, life cycle, okay, much more rapid than the cycle of humans or for farm animals. And so they can adapt to new situations. So in Asia, in particular in Thailand and in China, we had very severe diarrhea. And I was there at that time, and I can tell you it was very severe. The same severity as that you had in Mexico or they have in North America. and many herds were vaccinated against EU reference strain, the strain we had in Europe, with low effectiveness. So the case started in the USA in May, and then the rapid spread, and Mexico, you joined the club of infected countries, unfortunately, you can imagine. Next, I took these pictures in Thailand, and you have exactly the same here, okay? And the piglets, they look for warm, they look for heat, because they're totally dehydrated, they're cold. Crucial question, why did it emerge in the USA? So, many studies in many scientific areas and many hypotheses. The genome of many PD viruses were analyzed in depth and compared to other ones found around the world. And the analysis supported an Asian origin for the US outbreak. We are pretty sure of that. We are pretty sure that the virus came from Asia, somewhere in Asia. Okay? The question is, how could this virus come to the new continent? So, once again, there were a number of hypotheses and debate, and different scenarios were investigated. The role of feed ingredients, legally or illegally imported, was raised. And in particular, the question of SDPP, spray-dried porcine plasma. Okay? We know that with PED, there is a viremic phase. We also know that the virus can stay within the body of the pig many weeks after infection. So when the pigs are sent to the slaughterhouse, you take the blood to make the SDPP. The blood can be plasma more or less contaminated. Normally, the SDPP process can inactivate properly the virus. Okay? Does not resist to high temperature. The problem is that in the factories, to save money, to save time, there is a sort of variation in the level of the heat, of the temperature of drying process, and in the duration of the dwell time. So it might occur that in certain circumstances, in case of a highly contaminated raw material, and if the process is not carried on in the right way, that we could find virus, PED virus in SDPP. And that was the case in Canada, in Ontario. In these cases, the authorities went on the farms and in the feed mill, and in the feed mill they could find PED virus RNA in SDPP. So there are elements, at least there are proof of concept of the possibility of a transfer of virus from different places through the diet. You have to be careful of that. But the point is that it is not legal in the USA to buy SDPP from China. So if something came, it means that this was illegal. So, Nipah virus infection in Malaysia. This was a terrible case. 1 million pigs were killed and 100 people died. And we could reconstitute the things. And it came out that changes in local habitat of fruit bats through deforestation was the cause. So fruit bats, a colony of fruit bats, they are called flying foxes. They're very nice animals. Instead of keeping away in the forest, around at the border of the forest, close to a village, they came to live in the trees in the village, just above the piggeries. In Malaysia, the piggeries were partially roofed, so the bats went to stay there, to establish there, and the urine, the feces, and the fruit came down and contaminated the pigs and the people. Okay? Terrible. So, emergent in relation to pharmaceuticals and technologies. In 1984 in Europe, we have a pseudorabies vaccine infected with the pestivirus, was the virus of the border disease. 300,000 doses were used. Imagine that. The cause was that this company, the manufacturer, went to the slaughterhouse to catch, that was a way to cultivate the virus of Oyerski's disease, pseudorabies. He used kidney from sheep, okay? But unfortunately, one sheep was infected with border disease virus, okay? And they didn't know, of course. So this is an important point. Then live vaccine, PRRS live vaccine in Denmark in 1996. It gave PRRS symptoms and the virus could spread through the semen. And the comment I make here, care is needed with the live vaccine. You have to be very, very careful because they can recover virulence. So, side effect, collateral effect of pharmaceuticals, PCV2 replication after immunostimulation, and this was another accident, we will see that. This was an experiment we performed in our facilities. We tried to immunostimulate pigs and then to infect them. So first we immunostimulate, after that we infect them. So this is a complicated graph, but we will only focus on this group, the control group, and this black bars. So here is mentioned average daily gain. On average is 1 for the control. And immunostimulated first, then infected, you can see that you have a reduction in weight gain, reduction, and after that some of them recovered, but we have some mortality here. It means that when you immunostimulate young pigs, the pig will respond to that. And depending on this immune response, depending on the cells, depending on the material which are produced, the antibodies, the protein which are produced, the virus of PCV2, which is the chief opportunist, because it's a very small virus, it needs the help of the host cell machinery to replicate, okay? In this case, when you early stimulate in a certain way, we only use an adjuvant here, not a vaccine per se, only the adjuvant, not the antigenic part, okay? This generated specific cells, and PCV2 injected some days after took advantage and replicate, and we obtained PMWS, okay? So we have to be very careful, and sometimes trying to do better, the result is worst. So once again, you have to think about that. This was a problem that occurred 2007 in Germany and a little bit in Austria. It was caused by a vaccine due to control BVD, bovine viral diarrhea. Okay? And the incidence was rather low, 4,000 cases out of 3 million doses, but the mortality was high, 90% in the calves. And the problem was the presence of proteins of the MHC, the major histocompatibility complex, in the vaccine. And these proteins reacted with the blood cells and platelets of the serum of the calves. There was a development of alloantibodies that reacted that. And we had thrombocytopenia, we had leukopenia, and death very rapidly. The vaccine was removed in 2010. So what can be done? To sum up, the reasons mainly relate to human activities and to changing into microorganisms. So the viruses, the bacteria can change, but sometimes the changing are due to human activity, okay? So make the professionals really aware of the risk and their consequences. Education, quality control, safety, particular safety of pharmaceuticals, safety of the feed, We have some obstacles. Scientific knowledge is held in separate silos. We need, I think, to build bridges between the geneticists, the nutritionists, the veterinarians, make all these scientists work together and understand together each other. Human activities are driven by short-term economy to make profit, and to me, health in animal production goes hardly well along with short-term objective only. You need also mid-term and long-term. Priority is to be given to biosecurity and epidemiological. And the wildlife is not the problem as far as there is no contact. You have to avoid the contact and let wildlife live in peace. Preparedness roles official. Have action plans in case of a problem. And the conclusion, keywords in terms of prevention, biosecurity, doesn't work anymore, yeah. Biosecurity, avoid environmental disruption and proper hygiene and husbandry, prudent use of antibiotics, and the key point, over-reliance, I am not speaking about reliance, over-reliance on pharmaceuticals, including vaccine, at the expense of hygiene has to be discouraged. Every word is important in this sentence. And after that, ladies and gentlemen, I thank you for your attention.
Thank you. Perdón, antes de empezar con las preguntas, un anuncio rápido. Hay un Jetta blanco clásico, placas YKD 4299 con todas las luces prendidas, por si es de alguien de aquí que vaya a apagarlo, si no se va a quedar sin batería.
El tiempo es muy cruel y desgraciadamente no tenemos tiempo para preguntas, pero creo que fue una probadita. Sí, bueno, me dicen que sí, que se va a alargar el reloj. Hay dos preguntas, el doctor Becerril. Joaquín, ¿quieres preguntar en inglés? No, en español.
French, German, or English, my friend? or to me as well.
En italiano, por favor.
Or to me, or now what?
Pregunta en inglés.
Yes, okay, Dr. Madec, congratulations for your excellent talk. It's a privilege for all of us to have you here, such an outstanding researcher. Well, my question is actually regarding the last three most noxious diseases that we had in America and Mexico, to be talking particularly our country, are caused by RNA viruses. PRRS, swine influenza, and PED. So, how do you foresee for the next 10, 20 years, are we going to be prepared for another RNA disease? And the question is, we've been unable, all of us in the industry, along with the scientists, to develop effective vaccines to control this. How do you foresee? It's just a philosophical question, and again, congratulations. It's a privilege to have you here. Jorge, muchas gracias, felicidades al Comité Científico por traer a una eminencia de nivel mundial nada más. Muchas gracias.
Thank you for the point. Yes, of course, RNA viruses are unstable viruses, so they can change very rapidly, okay? But to me, new diseases cannot be predicted. We cannot predict that, okay? The disease can appear in another part of the world. Then it is appearing far from your country. Maybe that can leave time to you to prepare a vaccine and to be ready when the disease will arrive to your country. But to me, it is not predictable, actually new disease. So we have to be prepared in advance by good external biosecurity, because the pig is by far the best transporter of diseases, specific to pigs, like humans are for human disease, like cattle for cattle disease, and so on. Okay? And for sure, we have to work on vaccines and maybe develop new technologies specifically more adapted to RNA viruses, maybe. And we have good perspective in this work with the new technology, working on the sequence, of RNA, the new technologies. But we are also to be careful. We can also develop side effects, unpredictable at this stage at least. But we need action plans. We need, if necessary, to make an effort, of course, most effort on preparation of vaccines and biosecurity. That is my point.
Bueno, desafortunadamente el tema no nos permite más. El doctor va a estar esta mañana por aquí, entonces si quieren podemos abordarlo ya en privado para algunos otros cuestionamientos. Gracias de nuevo y un aplauso por favor para el doctor Madec.